and a warm welcome to this session of the Deep Dive. If your sights are set on acing the NEET exam, then you've definitely tuned in to the right place.
Yes, absolutely.
Today, we're getting right into the nitty-gritty of something super important: sexual reproduction in flowering plants. Trust me, understanding this properly, it can be a real game changer for your prep. We might even uncover some, fascinating points that'll help you remember everything more easily.
Absolutely. It's such a foundational topic, really essential for grasping plant biology, how traits get passed on.
Right.
And for NEET aspirants, a solid understanding of the structures, the sequence of events, and why they matter in the sexual reproduction of these angiosperms is just vital.
Right on. And to make sure we're building on the strongest base, we're doing a deep dive into the core concepts. We're looking at material based on some of the very first biology textbooks for higher secondary schools by NSERT, actually going back to 1964.
Oh, wow.
Yeah.
That's quite something.
Yeah. Compiled under the guidance of the esteemed Indian botanist Panchan and Maheshwari. So, we're looking at really reliable stuff here.
Indeed. Professor Maheshwari's work in those early NSERT textbooks, it was pivotal. Really shaped how biology was taught in India. They offered a very clear, very well-structured way to understand these fundamental life processes.
So our mission today is pretty simple, pretty focused. Break down the entire journey of sexual reproduction in flowering plants.
Okay.
We'll go from the initial structures right through to seeds and fruits, keeping a close eye on the key terms and, why each step is biologically significant. Think of it as supercharging your study notes with some extra insight.
Okay, ready to jump in.
Let's start at the very beginning, shall we? With the flower itself.
Hanan, the flower. It's such a captivating thing to look at, isn't it? We admire its beauty, the colors, the smells. It's there for, like, all our special occasions, prayers, even expressing feelings. But beyond all that, from a purely biological view, what's the main story? Precisely. While flowers, yes, they have huge aesthetic and cultural value, for us biologists, they are truly remarkable structures. They are the dedicated sites where sexual reproduction in angiosperms actually happens. Yeah. The sheer diversity we see, in how flowers are arranged on the plant, the individual flowers themselves, their different parts, it all reflects these incredible adaptations, all geared towards one thing: forming fruits and seeds. That's the ultimate outcome.
So it's all about making those fruits and seeds in the end. Now, even before we see a fully formed flower, there's a whole series of events happening behind the scenes, isn't there? Like things getting ready.
Absolutely. For a plant to even decide to flower, it involves a whole cascade of hormonal changes, structural changes, eventually leading to what we call the floral primordium.
Right.
This then develops into inflorescences, which bear the floral buds, and those mature into flowers. And inside these flowers, the male and female reproductive structures, the androecium and the gynoecium, they start to develop and differentiate.
Okay, let's start with the male reproductive part, the androecium. Who are the main players there?
Right, so the androecium is made up of a whorl of stamens. Each stamen is basically the male reproductive organ.
Okay.
A typical stamen has two main bits: a long, slender stalk called the filament, and a structure at the top, usually bilobed, which is the anther. The filament connects this anther to the thalamus, or sometimes to the petal itself.
And it's quite amazing how much these stamens can differ. Their number, their length, it can be so different from one type of plant to another. I remember just looking at different flowers shows you this huge variation.
Exactly. And if we take a closer look at that anther, especially if we cut across it transversely, we see it typically bilobed, and each lobe has two dikecas, so we call it dicticus. Often there's a groove running lengthwise that separates these dikecas. Inside the anther, it has this four-sided sort of tetragonal structure containing 4 microsporangia. They're located at the corners, 2 in each lobe. Think of it like a little 4-chambered box.
Microsporangia, right? That sounds like where all the male action happens. What goes on in there?
Well, these microsporangia, they develop further into what we call pollen sacs. These sacs run along the length of the anther and eventually become packed, absolutely filled with pollen grains. Now, if we zoom in on the structure of a single microsporangium, you'll see it's surrounded by 4 distinct wall layers.
4 layers. That's quite a bit of protection. Tell us about them.
Right. So from the outside moving in, you have the epidermis, then the endothecium, then the middle layers, and finally the innermost layer, the tapetum.
Oh, tapetum. Okay.
The outer 3 layers, their main job is protection. And they also help in dehiscence, that's the splitting open of the anther to release the pollen. But the tapetum, It has a very special, very crucial role.
Oh, yes, the tapetum. What's so special about it?
The tapetum is absolutely essential. It nourishes the developing pollen grains. Its cells have this dense cytoplasm, and often, interestingly, they contain more than one nucleus.
Multiple nuclei. Wow.
Yeah. Now, when the anther's still young, the very center of each microsporangium is filled with this group of tightly packed, similar cells. This is called a sporogenous tissue.
Sporogenous tissue. So that's where it all starts for the pollen. What do these cells do?
These cells, the sporogenous tissue cells, they are the ones that undergo meiosis, at the special type of cell division to form microspore tetrads. Each cell in this tissue is actually a potential pollen mother cell, or microspore mother cell. We call them PMCs.
Okay, PMCs.
And the process of forming these microspores from a PMC through meiosis is what we call microsporogenesis.
Meiosis, that's the division where the chromosome number gets halved, right? So the microspores in the tetrad, they're haploid.
Exactly right, haploid. And as the anthers mature and, dry out a bit, these microspores separate from each other in the tetrad, and each one develops into a pollen grain. Inside each microsporangium, you get thousands upon thousands of these microspores, or pollen grains, formed, all ready to be released when the anther finally splits open.
Okay, so we have thousands of pollen grains ready to go. What's their structure like? They just look like tiny dust particles usually, but they must have some pretty intricate design.
They really do. Pollen grains represent the male gamophytes. They're generally spherical, maybe 25 to 50 micrometers in diameter, and they have this very distinct two-layered wall.
2 layers, okay.
The outer layer, the exine, is made of something called sporopollenin. This stuff is incredible. It's one of the most resistant organic materials known. It can handle high temperatures, strong acids, alkalis. So far, we haven't found an enzyme that can actually break it down.
Wow. That's some seriously tough material. That must be what helps pollen survive out in the environment.
Absolutely. And this resistance is also why pollen grains are so well preserved in fossils. It gives us amazing clues about past plant life, past climates. That's the field of palynology.
Fascinating.
And the exine also has these prominent little openings called germ pores. Sporopollenin is absent there. These pores are crucial because that's where the pollen tube emerges later on during germination.
Right.
You might also notice the exine often have these really fascinating patterns and designs, which can actually help botanists identify different plant species, almost like a fingerprint.
Like a tiny fingerprint. And the inner layer, what's that like?
The inner wall is the intine. It's thin, it's continuous, and it's made up mainly of cellulose and pectin. Inside that, you have the cytoplasm of the pollen grain, enclosed by a plasma membrane. Now, when a pollen grain is mature, it typically contains 2 cells.
2 cells packed into that tiny sphere. How are they different?
There's the vegetative cell, which is the bigger one. It has plenty of food reserves stored up and a large, kind of irregularly shaped nucleus. Then there's the generative cell. It's smaller, and it actually sort of floats within the cytoplasm of the vegetative cell. It's usually spindle-shaped, with dense cytoplasm and its own nucleus.
Okay.
Now, here's a key point, especially for NEET prep. In over 60% of angiosperms, pollen grains are shed at this 2-celled stage with 1 vegetative and 1 generative cell.
Ah, okay, 60%.
But in the remaining species, that generative cell divides again, by mitosis this time, to form 2 male gametes before the pollen grain is even released. So in those plants, the pollen is shed at a 3-celled stage.
That's definitely an important detail to remember, 2-celled versus 3-celled stage. Now, pollen isn't always just about reproduction, is it? I remember reading about some other aspects, allergies, for instance.
You're absolutely right. Pollen grains from many species can be a real problem for some people, triggering severe allergies, respiratory issues like asthma, bronchitis.
Yeah.
A very common culprit, especially here in India, is parthenium, or carrot grass. It actually came in as a contaminant with imported wheat, and now it's everywhere, causing significant pollen allergy for lots of people.
Oh yes, parthenium allergy is quite common. But on the flip side, I also recall hearing about pollen being nutritious, like people eat it.
Absolutely. Pollen grains are packed with nutrients. And in recent years, there's been this growing trend of using pollen tablets as dietary supplements. In many Western countries, you can find all sorts of pollen products, tablets, syrups, marketed for their health benefits. Some even claim that consuming pollen could boost the performance of athletes and racehorses, though the evidence for that needs more study.
Interesting. So these tiny grains really pack a punch in both good and, not so good ways for some people. Now, once they're released from the anther, how long do they have? Do they need to reach the stigma quickly? Does pollen have a sort of best before date?
That's a really crucial question. Viability. Once shed, pollen grains have to land on the right stigma and germinate before they lose their ability to fertilize. And how long they remain viable? Well, it varies hugely.
How much variation are we talking?
A lot. It depends on the species and also on things like temperature and humidity. For example, in cereals like rice and wheat, pollen grains lose viability incredibly quickly within about 30 minutes of release.
Just 30 minutes. Wow, that's a tight deadline.
It really is. But then in some other plant families, like Rosaceae, roses, Leguminosae, peas, beans, and Solanaceae, tomatoes, potatoes, pollen can remain viable for several months.
That's a massive difference, months versus minutes.
Exactly. Think of rice pollen like a sprinter, and maybe rose pollen like a marathon runner. And just like we can store animal semen for artificial insemination, it's also possible to store pollen grains from many plant species for years. We do this in liquid nitrogen at a super low temperature of minus 196 degrees Celsius.
Minus 196.
Yeah. These stored pollen grains are kept in what we call pollen banks, similar to seed banks. They're incredibly useful resources for crop breeding programs, allowing breeders to make crosses even when plants aren't flowering at the same time.
That's a fantastic application of understanding pollen biology. Okay, we've thoroughly covered the male side, the androecium, and pollen. Now let's switch gears to the female reproductive part, the gynoecium. What's the basic setup there?
Right, the gynoecium represents the female part of the flower. It can be made up of just a single pistil, we call that monocarpellary, or it can have more than one pistil, which is multicarpellary.
Okay.
Now, if there are multiple pistils, they might be fused together. That condition is called syncarpous, like in hibiscus. Or they could be separate and free from each other, which we call apocarpous, like in a lotus or rose.
Syncarpous fused, epiocarpous free. Got it.
And each individual pistil, whether it's single or part of a fused group, typically has three distinct parts: the stigma, the style, and the ovary.
Stigma, style, ovary, the classic trio. What does each part do?
The stigma is usually the very tip of the pistil. It serves as the landing platform for the pollen grains. It's often sticky or might have hairs to help trap the pollen effectively. Makes sense. Below the stigma is the style, which is this elongated, slender stalk connecting the stigma down to the ovary. And the ovary itself is the basal, often swollen part of the pistil. Inside the ovary is the ovarian cavity, or locule. And within that cavity, attached to a sort of cushiony tissue called the placenta, are the ovules.
The placenta. Right, that's where the ovules are attached, like babies in a womb almost.
In a way, yes. These ovules are technically the megasporangia, and the number of ovules inside an ovary can vary dramatically between species. You might have just one ovule, like in wheat, paddy, or mango, or you could have many, many ovules, like in papaya, watermelon, or orchids.
From just one to hundreds or thousands, nature really covers all bases. Now let's look closer at the structure of these ovules, the megasporangia. What do they look like up close?
Okay, a typical angiosperm ovule is a small structure. It's attached to the placenta by a stalk called the funicle. The point where the main body of the ovule fuses with this funicle is called the hilum.
Funicle and hilum.
Then each ovule usually has one or two protective layers called integuments. These integuments surround a central mass of cells called the nucellus, but they don't completely enclose it. There's usually a small opening left at the tip, and that's called the micropyle.
Micropyle, like a little doorway.
Exactly. And at the opposite end from the micropyle, the base of the ovule, that region is called the chalaza.
Okay, so the nucellus is inside, protected by integuments, with the micropyle opening at one end and the chalaza at the base. What's inside the nucellus itself?
Within the nucellus, enclosed by the integuments, you find the embryo sac. This is the female gamophyte. The cells of the nucellus itself are packed with stored food material, providing nourishment for the developing embryo sac inside it. Typically, an ovule contains just one embryo sac, and this embryo sac develops from a single functional megaspore.
A megaspore. And how do these megaspores get formed? Is it similar to how microspores were formed?
Yes, the process is analogous. It's called megasporogenesis, the formation of megaspores from the megaspore mother cell, or MMC. Usually, a single MMC differentiates within the nucellus, typically near the micropylar region.
Okay, one MMC.
This MMC is a large cell, dense cytoplasm, prominent nucleus, and just like the PMC, it undergoes meiotic division.
Meiosis again, so having the chromosomes.
Right. This meiotic division results in the production of 4 megaspores, all haploid.
4 haploid megaspores. What happens to them? Do they all develop?
Here's a key difference from the male side. In the vast majority of flowering plants, only one of these 4 megaspores remains functional. The other 3 typically degenerate and just disappear.
Only one survives. Interesting.
Yes, and it's a single functional megaspore that goes on to develop into the female gametophyte, the embryo sac. Because it develops from just one megaspore, we call this type of development monosporic development.
Monosporic development makes sense. Okay, so this one functional megaspore, how does it transform into that complex embryo sac structure?
It's a fascinating process involving mitotic divisions, but only of the nucleus initially. First, the nucleus of the functional megaspore divides by mitosis to form 2 nuclei. These 2 nuclei then migrate to opposite ends, or poles, of the developing embryo sac. So now we have a 2-nucleate embryo sac.
Okay, one nucleus at each end.
Then 2 more rounds of mitosis happen sequentially at each pole. So the 2 nuclei become 4, and then the 4 become 8. This results in an 8-nucleate stage. It's like the blueprint being copied multiple times within the same space.
8 nuclei? That sounds like a crowded little cell.
It is quite remarkable. And a very important point here is that these initial mitotic divisions are free nuclear. That means the nucleus divides, but cell walls don't form immediately around each new nucleus.
Right.
They're all just sharing the same cytoplasm for a while.
Free nuclear divisions.
It's only after this 8-nucleate stage is reached that cell walls start to form, organizing these nuclei into distinct cells. This leads to the structure of the typical mature female gametophyte, the embryo sac.
And what's the final layout inside this mature embryo sac? It's often described in a very specific way, isn't it? 7 cells, 8 nuclei.
Yes, that's the classic structure for most angiosperms. 7-celled, 8-nucleate. There's a very characteristic arrangement. At the micropilar end, near that little opening, 3 cells group together to form the egg apparatus.
Egg apparatus?
This consists of one central egg cell, flanked by 2 supporting cells called synergids. These synergids have special cellular thickenings at their micropilar tips called the filiform apparatus. This apparatus looks like little fingers pointing inwards, and it plays a crucial role later in guiding the pollen tube towards the egg.
Guiding the pollen tube. Clever. What about the other end?
At the opposite end, the chalazal end, there are 3 other cells, typically just called the antipodals. Their exact function can vary, but they're part of the structure.
Okay, egg apparatus at one end, antipodals at the other. What's in the middle?
And right in the center, occupying the main volume of the embryo sac, is the large central cell. What's unique about this central cell is that it contains 2 nuclei, called the polar nuclei. They are typically located just below the egg apparatus before fertilization.
So we have the egg ready, helped by the synergids with their guides, the antipodals at the back, and this big central cell holding 2 polar nuclei. Quite a setup. Now that we've got both the male gametophyte, pollen, and the female gametophyte embryo sac fully developed, the next crucial step is getting them together. That's pollination, right?
Absolutely. You've hit the nail on the head. In flowering plants, both the male and female gametes are non-motile. They can't swim or move on their own, so they absolutely need a mechanism to bring them together for fertilization to happen.
And pollination is that mechanism.
Precisely. Pollination is simply the transfer of pollen grains, which are released from the anther, over to the stigma of a pistil. Flowering plants have evolved an absolutely amazing array of adaptations to achieve this transfer, often relying on external agents like wind, water, or animals to help out.
And these agents are so diverse, from simple wind and water to, a whole host of fascinating animals. Let's break down the different types of pollination first. What are the main categories?
Okay, based on the source of the pollen, where it comes from relative to the flower it lands on, we can broadly classify pollination into 3 main types: autogamy, geitonogamy, and xenogamy.
Autogamy first, that sounds like self-pollination, doesn't it? Pollen from the anther landing on the stigma of the very same flower.
Correct. Autogamy is self-pollination within the same flower. However, in a typical flower that opens up and exposes its anthers and stigma, we call those chasmogamous flowers, complete autogamy is actually quite rare.
Why is that?
Well, for it to work reliably, 2 conditions need to be met perfectly. The release of pollen and the receptivity of the stigma happen at exactly the same time, synchrony. And the anthers and stigma need to be positioned very close to each other so the pollen can easily land on it.
Right. Timing and position need to be just right.
Exactly. But some plants, like viola, common pansy, oxalis, and camelina, have evolved a really clever strategy to guarantee self-pollination. They actually produce 2 different types of flowers.
2 types?
Yes. They have the normal open chasmogamous flowers, but they also produce cleistogamous flowers. These ones are fascinating because they remain permanently closed. They never open at all.
Cleistogamous flowers, permanently closed. So how does pollination even happen inside a flower that never opens?
It's ingenious. Inside these closed cleistogamous flowers, the anthers and stigma lie very close to each other. When the anthers mature and split open, Dehea is inside the closed bud, the pollen grains just fall directly onto the stigma. So self-pollination is pretty much guaranteed.
Guaranteed pollination.
Yes, they are invariably autogamous because there's simply no chance for pollen from another flower to get in. This ensures that the plant produces seeds even if there are no pollinators around, which can be a big advantage in certain environments.
That sounds like a very reliable system for making seeds. But does relying only on self-pollination have any downsides for the plant in the long run? Maybe less variety?
That's a very insightful question. Yes, while cleistogamy ensures seed production, the downside is that continuous self-pollination leads to reduced genetic diversity over generations. This lack of variation can make the population less adaptable to changing conditions or diseases. It leads to what we call inbreeding depression.
Inbreeding depression.
Yeah.
Okay, that makes sense. Right, what's the next type? Geitonogamy. Sounds a bit similar.
Geitonogamy involves the transfer of pollen grains from the anther of one flower to the stigma of another flower, but on the same individual plant.
Ah, different flower, same plant.
Correct. So functionally, it looks like cross-pollination because it usually requires a pollinating agent like an insect or wind to move the pollen between flowers. However, genetically, it's very similar to autogamy because all the flowers on a single plant generally share the same genetic makeup. The pollen grains are coming from the same parent plant.
I see. So ecologically, it's cross-pollination, but genetically, it's like self-pollination. Okay. And the third type is xenogamy.
Yes, xenogamy is the transfer of pollen grains from the anther of a flower on one plant to the stigma of a flower on a completely different plant, but of the same species.
Different plant altogether.
Exactly. This is what we consider true cross-pollination. It's the only type that brings genetically different pollen grains to the stigma, which is crucial for promoting genetic variation within the population.
Right. This is where we get the real benefits of mixing genes. Now, let's talk about the helpers, the agents that make these transfers happen. We have the abiotic ones first, wind and water. How common are they?
Pollination by wind, which we call anemophily, is actually quite common among these abiotic methods. Wind-pollinated flowers have some characteristic features. They typically produce huge quantities of pollen grains.
Why so much?
Because wind pollination is very inefficient, it's a bit of a scattergun approach. So they need lots of pollen to increase the chances of some grains actually landing on a receptive stigma. The pollen itself is usually light, dry, and non-sticky, so it can be easily carried by air currents.
Okay.
The flowers often have well-exposed stamens, sometimes dangling out, so the wind can easily pick up the pollen. And the stigmas are often large and feathery to effectively trap that airborne pollen as it drifts by. Think of the tassels on a corn cob. Those silky threads are actually the stigmas and styles, perfectly designed to catch wind-blown pollen.
Ah, the corn tassels.
Right. Also, wind-pollinated flowers usually have only a single ovule in each ovary, and you often find numerous flowers packed together in an inflorescence. Grasses are another major group that relies heavily on wind pollination.
So it's a numbers game with wind pollination, lots of lightweight pollen, feathery stigmas. What about water pollination or hydrophily? Is that common?
Actually, pollination directly by water is relatively rare in flowering plants. It's limited to only about 30 genera, mostly monocots. You might think aquatic plants would commonly use water, but most of them, like water hyacinth or water lily, actually have their flowers emerge above the water surface.
Oh, so how are they pollinated?
They're typically pollinated by insects or wind, just like land plants. True water pollination occurs in plants like Vallisneria and Hydrilla, which grow in fresh water, and also in some marine seagrasses like Zostera.
How does it work in those cases?
It varies. In Vallisneria, it's quite neat. The female flower reaches the water surface on a long, coiled stalk. The male flowers release pollen grains onto the surface, and these are carried passively by water currents until they bump into a female flower's stigma.
Wow.
In seagrasses like Zostera, things happen underwater. Both male and female flowers remain submerged. The pollen grains are often long and ribbon-like, and they drift passively within the water until they contact a stigma.
Ribbon-like pollen.
Yeah. And in most of these water-pollinated species, the pollen grains have a special mucilaginous or slimy covering to protect them from getting waterlogged.
Makes sense.
And one more thing, both wind and water-pollinated flowers are generally not very colorful, and they don't produce nectar. There's no need to attract animal pollinators, you see.
Right. No need for flashy advertisements if you're relying on wind or water. That brings us neatly to the biotic agents, the animals. This is where things get really interesting with all those partnerships between flowers and their pollinators.
Indeed, the vast majority of flowering plants actually rely on animals for pollination, a process called zoophily. The range of animal pollinators is huge. Bees, butterflies, flies, beetles, wasps, ants, moths. You have birds like sunbirds and hummingbirds, and even bats in some cases.
So many different animals.
Absolutely. But among all these, insects, particularly bees, are the dominant biotic pollinating agents. They're incredibly important. And interestingly, even some larger animals have been documented as pollinators in certain regions. Things like lemurs, tree-dwelling rodents, even geckos and skinks.
Lizards pollinating flowers, it's amazing. What kind of adaptations do we see in flowers that rely on these animal helpers?
Well, often the flowers of animal-pollinated plants show very specific adaptations designed to attract their particular type of pollinator. For instance, flowers pollinated by bees and butterflies are often large, brightly colored, especially blue, purple, yellow, fragrant, and they produce nectar, a sugary liquid reward.
To reward.
Exactly. If the individual flowers are small, they're often clustered together into a conspicuous inflorescence to make them more noticeable. Flowers pollinated by flies and beetles, on the other hand, might actually produce foul odors, mimicking rotting flesh or dung to attract those specific insects.
Yeah, but effective.
Very effective. To ensure the animals keep visiting, the flowers usually provide these floral rewards. The main ones are nectar and pollen grains themselves, which are a good source of protein for insects. As the animal forages for its reward, its body inevitably comes into contact with the anthers and the stigma. Pollen grains in these animal-pollinated flowers are often sticky or have spines, so they adhere easily to the animal's body. When the animal then visits another flower of the same species, some of this pollen gets transferred to that flower's stigma, achieving pollination.
So it's a classic mutualism. The animal gets food, and the plant gets its pollen moved around. Sometimes the rewards can be quite unusual, though, right? Not just food.
Absolutely. In some fascinating cases, the reward isn't food, but a safe place for the animal to lay its eggs. A famous example is the enormous flower of Amorphophallus, sometimes called the corpse flower. It can be several feet tall. It emits this strong, unpleasant odor that attracts carrion beetles and flies. They come in, pollinate the flower while looking for a place to lay eggs, and the flower actually provides cavities for them to do just that. Another classic example is the relationship between the yucca plant and a specific species of yucca moth. The moth deliberately collects pollen, pollinates the yucca flower, and then lays its eggs inside the flower's ovary. The developing moth larvae then feed on some, but not all, of the developing seeds. It's a highly specialized codependent relationship.
Nature's partnerships are just incredible. Now, we talked about self-pollination and cross-pollination. Many plants seem to prefer cross-pollination. What about plants that really want to avoid self-pollination? They must have some strategies, some clever tricks up their floral sleeves.
You're absolutely right. Continuous self-pollination, as we mentioned, can lead to that inbreeding depression, reduced fitness, vigor, fertility. So flowering plants have evolved several clever mechanisms called outbreeding devices, specifically to discourage self-pollination and actively promote cross-pollination.
Okay, what are some of these devices?
One common strategy is simply ensuring that pollen release and stigma receptivity in the same flower are not synchronized. It can happen in two ways. Either the pollen is released before the stigma becomes receptive, that's called protandry, or the stigma becomes receptive before the pollen is released, that's protogeny. Either way, self-pollination is difficult.
A timing mismatch. Clever. What else?
Another mechanism involves the physical placement of the anthers and stigma. They might be positioned in such different locations within the flower that it's physically hard or even impossible for the pollen from the anther to land on its own stigma without help from a pollinator bringing pollen from elsewhere.
So physical separation within the flower. Got it.
A third very important device is self-incompatibility. This is a genetic mechanism. It basically prevents self-pollen from the same flower or even another flower on the same plant from successfully fertilizing the ovules.
How does it stop it?
It can work in a couple of ways. It might inhibit the pollen grain from even germinating on the stigma, or it might block the growth of the pollen tube as it tries to grow down through the style towards the ovary. It's a biochemical block.
A genetic lockout.
Exactly. And finally, perhaps the most effective way to ensure cross-pollination is the production of unisexual flowers. Flowers that are either only male or only female.
Right.
Now, if a plant bears both male and female flowers on the same individual, a condition called monoecious, like in castor or maize, It prevents autogamy, self-pollination within the same flower,
Yay.
but it doesn't prevent geitonogamy, pollination between different flowers on the same plant.
Ah,
But
okay.
in species like papaya, where male and female flowers are born on completely separate individual plants, it's called diatonosis, then both autogamy and geitonogamy are completely prevented. Cross-pollination is the only option.
Dionysi forces cross-pollination. That's a whole toolkit of strategies. Now, okay, let's say the right kind of pollen actually makes it to the stigma. It lands successfully. Is it smooth sailing from there, or is there more to it?
That's a very critical point. Just because pollen lands on the stigma, it doesn't automatically guarantee success. The pistil, the entire female reproductive unit, has this remarkable ability to recognize the pollen. It can distinguish between pollen that is compatible, the right type from the same species, and pollen that is incompatible from a different species, or sometimes even genetically incompatible pollen from the same species due to self-incompatibility mechanisms.
The pistil can tell the difference. How?
It's thought to be based on chemical signals, a kind of molecular conversation happening between the pollen grain and the surface of the stigma, and potentially the style tissue as well. If the pollen is recognized as compatible, the pistil accepts it and allows the next steps, germination and pollen tube growth, to proceed.
And if it's incompatible?
If it's recognized as incompatible, the pistil rejects it. This rejection might mean the pollen grain simply fails to germinate, or if it does germinate, the pollen tube growth might be arrested partway down the style. It's like the pistil has a very sophisticated security system checking credentials.
A biochemical gatekeeper. Amazing. So this recognition, this dialogue, it's crucial.
Absolutely. This ability of the pistil to recognize pollen and then accept or reject it is the result of this continuous chemical dialogue. Botanists are still actively researching the precise molecules involved in this complex recognition and response process.
And what happens when the pollen is accepted? When it gets the green light, what's the next step in this interaction?
Okay, so following successful compatible pollination, the pollen grain germinates on the stigma. A pollen tube starts to grow out from one of the germ pores we talked about earlier.
The pollen tube starts growing.
Yes, and the contents of the pollen grain, the vegetative nucleus and the generative cell, or the two male gametes if it was shed at the three-celled stage, move into this growing pollen tube. The tube then elongates, growing down through the tissues of the stigma and the style, heading towards the ovary.
It grows through the style tissue.
Yes, it essentially digests its way through the style tissue. Once it reaches the ovary, the pollen tube typically finds an ovule and enters it, usually through the micropyle, that little opening.
Through the micropyle doorway.
Exactly. And finally, it penetrates one of the synergids within the embryo sac. Remember the filiform apparatus in the synergids, those finger-like projections? They play a key role here, chemically guiding the tip of the pollen tube into the correct synergid cell.
So the synergids help guide it in.
Precisely. All these events, starting from the pollen landing on the stigma, its germination, the tube growth down the style, entry into the ovule, and finally reaching the embryo sac, this entire sequence is collectively referred to as the pollen-pistil interaction.
Pollen-pistil interaction.
Understanding this whole dynamic process is incredibly valuable, especially for plant breeders. It allows them to sometimes overcome natural incompatibility barriers and make successful crosses between plants that might not normally hybridize, leading to desired new crop varieties through artificial hybridization.
Artificial hybridization. Right, that sounds like how breeders create new types of plants, giving nature a bit of a helping hand. How exactly is that done?
It's a cornerstone technique in crop improvement. Plant breeders intentionally cross-pollinate different species, or maybe different varieties within a species, to combine desirable traits like higher yield, disease resistance, better flavor, into one superior offspring plant. It often involves very careful procedures, mainly emasculation and bagging.
Emasculation and bagging. What are those?
Okay, if the plant you want to use as the female parent has bisexual flowers, meaning flowers with both stamens and pistils, you have to prevent self-pollination. So before the flower opens fully and the anthers mature, you carefully remove the anthers using forceps. That removal of anthers is called emasculation.
Removing the male parts.
Exactly. Then the emasculated flower is immediately covered with a bag, usually made of butter paper or something similar, to prevent any unwanted stray pollen from landing on the stigma. This is bagging.
Protecting it.
Right. Then, when the stigma of this bagged flower becomes receptive, the breeder takes mature pollen grains that have been collected from the desired male parent plant and carefully dusts them onto the stigma. After this controlled pollination, the flower is immediately rebagged to allow the fruit and seeds to develop without any further uncontrolled pollination.
Very meticulous work.
It has to be precise. Now, if the female parent plant naturally produces only unisexual female flowers, then obviously emasculation isn't needed. You just need to bag the female flower bud before it opens, pollinate it with the desired pollen when ready, and then rebag it.
So it's a very controlled way to ensure only the desired cross happens. Now, after all this prep, pollen delivery, interaction, we finally arrive at the main event, fertilization itself. And in flowering plants, it's famously not just a single event, it's a kind of double feature, isn't it?
That's absolutely right, double fertilization. It's one of the defining characteristics of angiosperms. Here's what happens. After the pollen tube enters the cytoplasm of one of the synergids and ruptures, it releases the 2 male gametes it was carrying.
2 male gametes released.
Yes, one of these male gametes travels towards the egg cell and fuses with the egg cell nucleus. This fusion of a male gamete with the egg nucleus is called xenogamy, and the result of this fusion is the formation of a diploid cell, the zygote. This zygote contains genetic material from both parents and has the potential to develop into the embryo.
Xenogamy forms the zygote. Okay, what about the second male gamete?
The other male gamete moves towards the large central cell, the one containing the 2 polar nuclei. This second male gamete fuses with both of the polar nuclei simultaneously.
Fuses with 2 nuclei at once.
Yes. Since this fusion involves 3 haploid nuclei, 1 male gamete nucleus plus 2 polar nuclei, it's called triple fusion. The result of triple fusion is the formation of a triploid nucleus, which we call the primary endosperm nucleus, or PEN.
Triple fusion forms the PN, triploid.
Correct. So you have these 2 distinct fusion events happening almost simultaneously within the embryo sac. Xenogamy, male gamete plus egg diploid zygote, and triple fusion, second male gamete plus 2 polar nuclei triploid PEN. This combination of both events occurring together is what constitutes double fertilization. It is completely unique to flowering plants.
Double fertilization, xenogamy, and triple fusion. Truly unique. And what happens after these fusions? What do the zygote and the PEN become?
Right after double fertilization, the central cell, now containing the triploid PEN, is referred to as the primary endosperm cell, PEC. This PEC will divide and develop into the endosperm, which, as we'll see, is a nutrient-rich tissue designed to nourish the developing embryo. Meanwhile, the diploid zygote will undergo development to form the embryo itself, the miniature future plant.
So one fusion creates the baby plant embryo, and the other creates its food supply, endosperm. Ingenious. Now, after this double event, there's a whole cascade of post-fertilization changes that happen in the ovule and ovary, isn't there?
Yes, indeed. Following double fertilization, a series of significant events unfolds. We see the development of the endosperm, the development of the embryo from the zygote, the maturation of the ovule into seeds, and the transformation of the ovary into a fruit. These are all collectively known as post-fertilization events.
Okay, let's tackle them one by one. Starting with the endosperm. We just said it provides nourishment. How does it actually develop from that primary endosperm cell?
Endosperm development usually gets a head start. It typically begins before the embryo starts developing significantly. This makes perfect biological sense. You need to have the food ready before the baby starts growing rapidly.
Prepare the pantry first.
Exactly. The primary endosperm cell, PEC, undergoes repeated mitotic divisions, forming the triploid endosperm tissue. The cells of this tissue become packed with stored food materials, starch, proteins, oils, depending on the species, which will fuel the embryo's growth.
How does the division happen? Is it always the same way?
The most common method of endosperm development is called free nuclear endosperm formation. In this type, the primary endosperm nucleus, PEN, undergoes successive rounds of nuclear division, but without immediate cell wall formation. So you end up with potentially hundreds or thousands of free nuclei suspended in the cytoplasm of the central cell.
Just free nuclei floating around?
Initially, yes. It's only later that cell walls begin to form around these nuclei, starting usually from the periphery and moving inwards, eventually making the endosperm tissue cellular. The extent of this free nuclear phase can vary a lot between different plants. A classic, very familiar example is the coconut.
Ah, the coconut. How does that fit in?
The liquid inside a tender green coconut, the coconut water, is actually the free nuclear endosperm, full of those free nuclei. The white kernel, the fleshy part that we eat, that's the cellular endosperm that forms later as cell walls are laid down.
Wow, I never knew that. Coconut water is free nuclear endosperm. Fascinating.
Yeah.
So what ultimately happens to this endosperm? Does it always stick around in the final seed?
Not necessarily. In many plants, the endosperm gets completely used up by the developing embryo before the seed even reaches maturity. This happens in seeds like peas, groundnuts, and beans. We call these non-albuminous or X-albuminous seeds. In these cases, the food reserves needed for germination are stored directly in the embryo's cotyledons, which become thick and fleshy.
So the embryo eats all its packed lunch before the journey starts.
You could put it that way. Alternatively, in other plants, the endosperm persists in the mature seed. These are called albuminous seeds, like in castor, coconut, and most cereals, wheat, rice, maize. In these seeds, the endosperm serves as the food source during seed germination, nourishing the young seedling until it can establish its roots and leaves and start photosynthesizing.
Albuminous seeds keep the lunchbox packed for later use.
Precisely. And sometimes there's even another leftover. In some seeds, like black pepper and beet, remnants of the nucellus, the tissue that originally surrounded the embryo sac, also persist in the mature seed. This persistent nucellus is called the perisperm, and it can also act as a storage tissue.
Perisperm. Extra snack. Okay, so that's the food supply sorted. Now, what about the main event, the embryo itself? How does it develop from that single-celled zygote?
Embryo development, or embryogeny, typically starts at the micropylar end of the embryo sac, which is where the zygote is located. Interestingly, the zygote usually doesn't start dividing immediately after fertilization. It often waits until at least some amount of endosperm is formed, ensuring there's a nutrient supply ready.
Smart move.
And although the final mature embryos in monocots and dicots look quite different, the very early stages of their development embryogeny are surprisingly similar across both groups. The zygote divides repeatedly, forming a sequence of structures. It progresses through stages, often described visually as the proembryo, then a globular stage, then a heart-shaped stage in dicots, and finally maturing into the fully formed embryo.
Globular, heart-shaped. Okay, let's look at a typical dicot embryo first, like in bean seed. What are its key parts?
Right. A typical dicot embryo consists of an embryonal axis and two prominent cotyledons, the seed leaves. The part of the embryonal axis that lies above the point where the cotyledons are attached is called the epicotyl. It terminates in the plumule, which is the precursor to the shoot system, the future stem and leaves.
Epicotyl and plumule for the shoot. What about below the cotyledons?
The cylindrical part of the axis below the level of the cotyledons is the hypocotyl. It terminates at its lower end in the radicle, which is the embryonic root, the future root system. And the very tip of the radicle is usually covered and protected by a structure called the root cap.
Hypocotyl and radicle for the root. Simple enough. Now, how does a monocot embryo, say in a grain of maize or wheat, differ from this structure?
The most obvious difference is that monocot embryos possess only one cotyledon. In the grass family, which includes cereals like maize and wheat, this single cotyledon is large, shield-shaped, and called the scutellum. It's positioned laterally, sort of pressed against the side of the embryonal axis.
One cotyledon called the scutellum.
At the lower end of the embryonal axis, you still have a radicle and the root cap, but they're enclosed within an undifferentiated protective sheath called the coleorrhiza.
Coleorrhiza protecting the radicle.
Yes, and the portion of the embryonal axis above the attachment point of the scutellum is the epicotyl. This contains the shoot apex and a few young folded leaf structures called leaf primordia. And this whole shoot apex region is enclosed in another protective, hollow, sheath-like structure called the coleoptile.
Coleoptile protecting the shoot apex. So key differences are one cotyledon scutellum and these protective sheaths, coleorrhiza and coleoptile.
Exactly. Those are the main distinguishing features of a monocot embryo, particularly in grasses.
Right. Now, as the embryo develops, the entire ovule is maturing into a seed. What's the basic structure of a typical seed?
Okay, the seed is the final product of sexual reproduction in these plants. It's essentially a mature, fertilized ovule, ready for dispersal and germination. A typical seed consists of one or more protective seed coats, the cotyledon which store food or help transfer it, and the embryo axis itself.
Seed coats. Where do they come from?
The seed coats develop from the integuments, those protective layers that surrounded the ovule. As the seed matures, the integuments harden and become the tough outer layers, providing physical protection to the embryo inside. The micropyle, that small opening in the ovule, often persists as a tiny pore in the seed coat. It's important because it allows oxygen and water to enter when the seed starts to germinate.
Ah, the micropyle still has a job.
Yes. As the seed matures, its water content is significantly reduced, typically down to about 10–15% moisture by mass. It becomes relatively dry. This dehydration slows down the metabolic activity of the embryo considerably, and the embryo may enter a state of dormancy or inactivity, waiting for favorable conditions like adequate moisture, oxygen, and suitable temperature before it germinates.
Dormancy. like suspended animation. And while the ovule turns into a seed, something's happening to the ovary too, right? That becomes the fruit.
Yes, exactly. The processes happen more or less simultaneously. As the ovules mature into seeds, the ovary surrounding them develops and ripens into the fruit. The wall of the ovary thickens and differentiates to become the fruit wall, which is technically called the pericarp.
Pericarp, the fruit wall.
Right. The pericarp can be quite variable. It might be fleshy and succulent, like in fruits such as guava, orange, mango, or it can be dry, like in groundnut or mustard pods. Many fruits have also evolved diverse and often ingenious mechanisms to help disperse the seeds contained within them using wind, water, animals, or even explosive mechanisms.
Seed dispersal, another fascinating area.
Definitely. It's also worth noting the relationship between the ovary and the fruit. Generally, fruits that develop solely from the ovary are called true fruits. But in some plants, other floral parts like the thalamus, the base of the flower, also contribute significantly to the structure we eat as the fruit. Examples include apple, strawberry, and cashew. Yeah, we call these false fruits or accessory fruits.
Apples and strawberries are false fruits. Interesting.
Yes, the main fleshy part we eat in an apple is actually derived from the swollen thalamus, not the ovary wall. And then there's another category, parthenocarpic fruits. These are fruits that develop without the process of fertilization ever happening. Because there's no fertilization, they're typically seedless. Banana is a common natural example. Parthenocarpy can sometimes also be induced artificially in other foods by applying certain plant growth hormones to the unfertilized ovary.
Seedless fruits like bananas.
Yeah.
Parthenocarpy. Got it. So seeds and fruits, the grand finale of this whole intricate reproductive process, they are obviously vital for the plants themselves, ensuring the next generation. What makes seeds in particular so biologically significant? Why was evolving the seed such a big deal?
Seeds offer several major advantages to angiosperms, contributing hugely to their success. Firstly, unlike reproduction in many lower plant groups like ferns or mosses that need water for fertilization, seed formation is independent of external water for the final fusion of gametes. This makes sexual reproduction much more reliable and dependable, especially in drier terrestrial environments.
Reliability, okay. What else?
Seeds are also fantastic dispersal units. They have evolved countless ways to travel far from the parent plant, carried by wind, water, animals allowing plants to colonize new habitats and avoid competing with their own parent.
Better dispersal. Makes sense.
Then there's the stored food. The endosperm, or the cotyledons, provide essential nourishment to the young seedling during germination, giving it a head start until it can establish itself and begin making its own food through photosynthesis.
Built-in food supply.
The hard seed coat provides excellent protection to the vulnerable embryo inside, shielding it from physical damage, dehydration, and even pathogens. And crucially, because seeds are the product of sexual reproduction involving meiosis and fertilization, they generate new genetic combinations, leading to variation within the population. This variation is the raw material for adaptation and evolution.
Protection and genetic variation.
Absolutely. And from a human perspective, seeds are the very foundation of agriculture. Their ability to be stored for long periods allows us to save crops from one season to plant in the next, forming the basis of our food supply.
Couldn't agree more. Agriculture revolves around seeds, and the ability of some seeds to remain viable, to stay alive for incredibly long periods. That's just astonishing, isn't it?
It truly is mind-blowing. While some seeds, like we said for pollen, lose viability quickly, maybe within a few months or a year, others can remain alive for decades, centuries, or even millennia under the right conditions. There are some famous examples.
Like what?
Well, there's the case of the Arctic lupine, Lupinus arcticus. Seeds excavated from Arctic tundra, estimated to be around 10,000 years old, were successfully germinated.
10,000 years. Incredible.
And another famous one is a date palm seed, given the name Methuselah, which was discovered during an archaeological excavation at Masada near the Dead Sea. It was dated to be approximately 2,000 years old, and incredibly, it was successfully sprouted and grown into a plant.
Wow, 2,000 years old. That's just unbelievable resilience packed into a tiny seed. And when you think about the sheer number of seeds some plants can produce, the reproductive capacity is staggering sometimes.
It really is. Just consider the potential multiplication. The number of ovules in an ovary, the number of ovaries in a flower, the number of flowers on a plant. It adds up. Some plants take it to extremes. Orchid fruits, for example, are capsules that can contain hundreds of thousands, sometimes even millions, of tiny dust-like seeds.
Millions from one fruit.
Yes. Similarly, some parasitic plants like Orobunch and Striga also produce enormous numbers of very small seeds to maximize their chances of finding a host. And perhaps the most dramatic example is thinking about a giant ficus tree, like a banyan. It starts from a single tiny seed, but over its lifetime it can grow into a massive tree, producing potentially billions of seeds. It's a powerful testament to the effectiveness of this whole reproductive strategy.
Billions. It really puts the power of reproduction into perspective. Now, towards the end here, there are a couple of interesting variations or deviations from the standard sexual reproduction pathway we've discussed. Apomixis and polyembryony. What are these phenomena all about?
Right, these are fascinating exceptions to the rule. Apomixis is a term used to describe a situation where some flowering plants is particularly common in families like the Asteraceae sunflower family, and grasses have evolved a way to produce seeds without involving fertilization.
Seeds without fertilization, how does that work?
It's essentially a form of asexual reproduction that cleverly mimics sexual reproduction because it still results in a seed. There are a few different biological pathways. In some cases, the egg cell itself develops from a diploid cell, formed without meiosis, the reduction division, and this diploid egg then develops directly into an embryo without ever being fertilized.
So the egg just develops on its own.
Yes, parthenogenetically, but starting from a diploid cell. In other common cases, particularly in plants like citrus, oranges, lemons, and mango, some of the cells in the nucellus, remember, the tissue surrounding the embryo sac, which are diploid, start dividing unexpectedly. They protrude into the embryo sac and then develop into embryos themselves.
Embryos from the nucellus tissue, wow.
Yes, so in apomixis, because there's no fusion of gametes from two parents, no meiosis or xenogamy in the formation of the embryo, the resulting seeds produce offspring that are genetically identical to the mother plant. They are essentially clones.
Clones produced via seeds. And what about polyembryony? That sounds like many embryos.
Exactly. Polyembryony is simply the occurrence of more than one embryo within a single seed. This often happens as a result of apomixis, especially the type where those nucellar cells form extra embryos. So in many citrus and mango seeds, you might find the normal sexually produced zygotic embryo, plus several additional apomictic embryos derived from the nucellar tissue, all packed into the same seed.
Ah, so that's why sometimes when you squeeze an orange seed, you see multiple little sprites inside?
Precisely. That's polyembryony, often linked to apomixis in citrus.
Yeah.
You have the real embryo from fertilization, and then these bonuses clonal embryos from the nucellus.
Apomixis and polyembryony. Really interesting biological quirks. What's the potential significance of apomixis, particularly thinking about agriculture and maybe the hybrid seed industry?
Apomix phoenix holds tremendous potential, yes.
Mm.
Hybrid crop varieties, as you know, often show significantly higher yields and better performance due to hybrid vigor. But a major challenge is that farmers usually have to buy fresh hybrid seeds every single planting season.
Why is that?
Because if you collect seeds from a hybrid plant and sow them, the desirable traits segregate out in the next generation due to meiosis and genetic recombination. The offspring won't be uniformly vigorous like the original hybrid. Producing new hybrid seeds each year is also a costly and labor-intensive process, making the seeds expensive for farmers.
Okay, so hybrid traits get diluted. How would apomixis help?
Well, imagine if you could make a hybrid variety that was also apomictic. The hybrid embryo could still be produced initially through a normal cross, but then the plant could produce seeds apomictically. These apomictic seeds would contain embryos that are genetically identical clones of the superior hybrid parent plant.
So the hybrid traits would be locked in.
Exactly. Farmers could then save and reuse these apomictic hybrid seeds year after year, generation after generation, without losing the hybrid vigor or other desirable characteristics. This could dramatically reduce the cost of cultivation and significantly boost agricultural productivity, especially for smallholder farmers.
That sounds like a potential revolution for agriculture.
It really could be. Because of this immense practical importance, there's a huge amount of research going on worldwide to understand the genes that control apomixis, and to figure out how we might be able to transfer these genes into important hybrid crop varieties like maize, rice, and vegetables. It's a major goal in plant breeding.
A very exciting prospect indeed. Well, we have covered an incredible amount of ground today. Seriously comprehensive. From the basic structure of the flower as the stage for reproduction, all the way through pollination, that unique double fertilization, the development of the endosperm, embryo, seed, and fruit. And even these fascinating side routes like apomixis.
Yes, it's been quite a journey. We've looked at the pre-fertilization structures and events, both male and female, the amazing mechanisms plants use for pollination, that defining double fertilization event, and all the crucial post-fertilization developments. It really is a complex but beautifully orchestrated series of biological processes.
And for all of you listening, especially those diligently preparing for your NEET exam, really getting a solid grip on these fundamental concepts is absolutely key. It builds such a strong foundation, not just for cracking this exam, but also for your future studies in botany, genetics, and even as you move toward your medical aspirations eventually.
Hmm.
So maybe take some time after this to really think about the intricate steps we've discussed, the journey of the pollen grain, the development inside the ovule, the formation of the seed, and reflect on their profound significance, both in the natural world all around us and in the food that sustains us every day.
Indeed. And perhaps as you continue your studies, mull over a couple of broader questions that arise from this. For instance, considering this vast diversity of reproductive strategies we saw in flowering plants, wind, water, insects, self-pollination, cross-pollination, apomixis, what specific evolutionary advantages might these different approaches offer plants living in different kinds of ecological niches or facing different environmental challenges?
Oh, good question.
Or thinking of the future, how might further breakthroughs in our understanding and application of apomixis genuinely revolutionize agricultural practices and significantly contribute to enhancing food security, particularly in a country like India, with its vast population and reliance on agriculture? These are the kinds of thought-provoking questions that build upon the core knowledge. We really encourage you to explore them further.
Excellent food for thought to take away. Thank you so much for joining us on this deep dive today. We sincerely hope this session has given you a clearer, perhaps more engaging, and ultimately more useful understanding of this vital topic, sexual reproduction in flowering plants. We wish you all the very best with your NEET preparations.